Okay. Hello. Hi, everyone. Thank you for being here this afternoon. Lovely weather outside. No, it's nice to have you here now. So before we start, I'll introduce myself. I'm Jesus Aguirre. I'm an associate professor on biodiversity and earth observation here at SOGI. And today I will actually I'm hosting Cristina Banks-Leite from Imperial College London. But before I just talk a bit about her, I'll just make a couple of announcements. So a couple of the talks that we will have in the next 2 weeks. The first one on Friday, the 21st of November. It is with Syrian Sumner from the University College London, talking about the natural capital of wasps, followed by a book signing of her book, Endless Forms, The Secret World of Wasps. And then on Friday the 28th, we will have Rosa Vasquez Espinosa from Amazon Research International. And she will be speaking about the spirit of the rainforest, also followed by a book signing of her book, The Spirit of the Rainforest. So if you can make it in the next 2 weeks, I think it's going to be super exciting. And also, before we start, just to remind you that we will have some drinks reception for Chris after the talk in the atmosphere room, which is just around the corner. Okay, for Cristina Banks-Leite is a professor of biodiversity change in the Department of Life Sciences at Imperial College London. Her research focuses on measuring, understanding, and mitigating biodiversity loss across tropical and urban ecosystems. She integrates technologies such as bioacoustics, AI, and molecular tools to monitor biodiversity in novel ways. Cristina leads long-term collaborations with organizations including reforestation companies in Brazil and the City of London Corporation, applying her expertise to real-world conservation challenges. Her work has influenced policy in multiple countries, including legislation in Brazil and infrastructure planning in Costa Rica, and has been cited in over 40 policy documents worldwide. She has received multiple awards and was named one of the most influential female researchers in the UK. Cristina, thank you for being here.
Thank you so
The
much.
floor is yours.
Thank you. Hopefully you can hear me. Thank you so much, Jesus, for the invitation. It's a pleasure to be here. It would have been nice if it was sunny, but it's okay. It's okay. I'll forgive you, Jesus. So yes, I'm from Imperial College, and you can see that this slide is already quite outdated. Imperial is not called Imperial College London anymore. Silwood Park has had its 75th anniversary quite a few years ago, but it's such a pretty slide, I thought I'd continue using it. So today I'm going to be talking about what drives species sensitivity to deforestation. And I just wanted to go back a little bit to what Jesus had said. So my line of my research is really focusing on these three types of topics, kind of measuring biodiversity change. So I've got this collaboration with a company in Brazil called Mombach, which I just recorded a podcast about. So if you're interested, do go and have a look. And I'll be happy to talk about this. I'm also more than happy to talk about how the work I've done with this idea of mitigating biodiversity change, how we can reduce it. But today, really, I'm just going to focus on the understanding the drivers. So when we look around this room, we can see that people vary in all sorts of different ways. So we've got very different shapes and sizes and etc. And we know that if we go into a cold place, we're going to have slightly different requirements than if we go to a hotter place. So for instance, if we go to, if we go here at the mountains, the snowy mountains, you're going to have to be in a house that's quite warm. You're going to need kind of, you're going to adapt the environment to you. That's what humans do. If you go out to this lovely place, I don't know where it is, but you'd go out into the sea and kind of try to refresh yourself. So we understand this very easily for humans. It's, it's something we don't even have to think twice about it. Now, when it comes to other species, we very easily forget about it. So they probably it's because they all look the same. We can't really tell the difference between different individuals. So we just kind of objectify them to some extent, but it's also obvious for us that, you know, intraspecific variation is super important. We know that it drives speciation, it's, you know, behind evolution and all of that. Everyone knows that. But when it comes to coming up with the conservation decisions, we treat those species almost all of the time as a single unit, just the, you know, each different species. And this idea of kind of treating them in a kind of unit way and thinking of them as kind of responding to the environment in the same way wherever place they are is something that is so ingrained in our culture. Since the ancient Greeks, we've had this idea of indicator species, that, you know, an indicator species is always going to indicate a certain condition wherever that species is found. And so I've kind of also been, you know, guilty of that to some level. Like when I started my PhD, one of the main chapters for my thesis was to create a set of indicator species for the region that we were working. And so one of the things that I did was to identify indicator species, but also identify landscape metrics that could indicate the changes in biodiversity. And much to my surprise, what we found was that landscape-based indicators, so kind of metrics of the environment, when we had several different landscapes, several different regions that we had this data from. So what we tried to do was identify the indicators in one place and then apply to another place to see whether they would be transferable, right? So what we found was that when we tested for this transferability, the landscape-based indicators, they maintain their explanatory powers. This means that if I say that forest cover is the single most important indicator, let's say, and I do this for this region here, but then I apply it in another region, I still have a very strong predictor ability for that indicator. But when we found, let's say, species A, B, C for one place, that those species would be great indicators, and we applied them for another place, what we found is that the effectiveness of their predictive ability reduced by 33%. So that was a little bit odd to me at the time. I didn't know exactly what to do with those results. But later on, Jack Hatfield, who was my first PhD student, he looked into traits. So we all know that traits are, you know, species all have their, you know, very specific traits. And there's a very large body of literature showing that species with certain particular traits are more likely to be sensitive to land use change than species with other types of traits. So Jack had two different data sets. But these data sets were slightly different because they were collected in exactly the same region, but by two different people. So some of the sites were exactly the same. Most of the species were exactly the same, but they were collected by two different people. So they were just slightly different. So what he did was, he again looked at the traits that best explain species sensitivity in one data set, and then applied to the other data set. Can we still identify the species that are going to be most sensitive? And again, we didn't find that. So this is a part of the result, the abstract, where we say that our results demonstrate that the use of species traits to predict sensitivity to anthropogenic habitat loss can produce predictions that are species and site specific and not scalable to whole regions or biomes, and thus should be used with caution. So this was something that, again, it's kind of like bubbling in my mind. And I didn't know exactly what to do with this. And all around this kind of same time, this was actually happened when I was still kind of finishing my PhD. So I did my PhD on birds in the Atlantic forest of Brazil, near the region of São Paulo. And I met a guy from Argentina who had done a very similar type of work, exactly similar questions, exactly same thing, but in Argentina, right? So as you do, you know, talking to him, oh, species X, oh, so sensitive. And he's like, no, it's not. And then I said, well, species Y is really, that one is everywhere. And it's like, no, it's the opposite. So we try to kind of put together these results and say, okay, so if we were to measure species sensitivity in Brazil and in Argentina for the same species, would they have exactly the same levels in the two different areas? So imagine here we've got, we've got two different species here, one that it's benefited by habitat loss, another one that it's sensitive to habitat loss. And imagine you measure species sensitivity in whatever ways. There's several different ways. I'm happy to talk about how we've been measuring species sensitivity later. But basically, if the idea that species have one single response, all populations within a species have one single response to habitat loss, then we would expect, this bird that's have very low sensitivity would be here both in Argentina and in Brazil, and a bird that has very high sensitivity would be here both in Argentina and Brazil. You'd get pretty much like, ideally you'd get a one-to-one relationship, right? But this is kind of what we found. It wasn't that, but this is pretty much what we found. There was absolutely no relationship at all. So all of this was happening, and I was thinking, what's going on here? I absolutely cannot put these results into context. So around the same time as well, we started looking into lots of different reasons for why species, populations within a single species would respond to habitat loss in different ways in different places. So one of the hypotheses, one of the factors that we looked into was the center periphery hypothesis. So this is a slightly controversial hypothesis that's been out for quite a long time. There's a very good review by Samuel Piranon in 2017, if you're interested. And the idea is that, so all species occur within, can you see the pointer? Good. So all species occur within a range, and populations that are closer to the center of this range are likely to have higher abundance, they're likely to have higher genetic diversity, whilst populations that occur kind of towards the edge of this range are likely to be more, smaller, just because there are fewer individuals coming from all of these areas, which is called mass effects, and there's less dispersal from other areas, and they're just in an area that's just slightly uncomfortable for them, right? It's just a little bit too hot, a little bit too cold. Something's going on for them. So we decided to put this to the test. So we wondered whether populations located at the range edge are more sensitive to habitat loss than those within the range core. So this was, so for that, at the time, we had 5 data sets from the Atlantic forests of Brazil. So this is South America, this is pretty much the distribution of the Atlantic forest in Brazil. And as you can see, it's a highly deforested biome, because what's black, it's where forest is, and the rest is just non-forest. So it's pretty bad, the situation there. But anyway, we had 5 data sets on the same, again, just on birds, but they were using the same methods, all by really good researchers that we knew they had really good, they had done this work really well. So the work was done by Sarah Mayer, who was a master's student at the time. Sarah went on to do her PhD in Zurich. And then David Orme took it over, and then they published jointly as first authors. So the first thing we did was to say, okay, so let's go back to my experience with the Argentinian guy, right? So is there a correlation in species sensitivity in the different data sets? And this is what we found. So as you can see, it's pretty bad. So I don't know if those at the back can see the numbers, but we're talking about R-squareds of 0.04, a negative relationship, but of 0.07. So it's really, really bad. The strongest one is 0.216, which is a bit meh, right? It's not great. So it kind of corroborated the idea that something's going on. Whatever species are not, populations from the same species are not responding to habitat loss in the same way across their range. So this is a very complicated graph, which I'm going to go a little bit in detail about it to explain. So what we did first was to choose the 25 species that occurred in all 5 studies. And then we measured their sensitivity to habitat loss. Again, I'll explain later if you're interested. And we looked to see how their sensitivity varied from distance to the range edge. Yes, some populations occur outside the range. It happens. So it went all the way from kind of minus 100 kilometers to 2,500 kilometers. And so here we've got 25 times 5 points. And each of these lines are for each of the different species. And as you can see, for the vast majority of lines, there is a very steep decline where basically species have really high sensitivity to habitat loss when they're located close to the range edge, but quite lower sensitivity. The populations are much lower sensitivity when they're located away from the range edge. There are a few exceptions. So for instance, this trogon here, it's a species that we only found up to 400 kilometers from the range edge. This species was always sensitive. So species that have small ranges are likely to be always close to the range edge. But kind of like, look, if we are to build in these results in a slightly more the way that people usually see, so kind of looking at how the incidence, from 0 to 1 changes. So for instance, here we can see that this woodpecker here, for instance, close to the edge, we can see the species having quite a strong positive response to forest cover. So species that we would consider very, very sensitive. But then when we find the species far from the range edge, the species reduces in incidence with increasing forest cover. So basically, you have all of these types of responses here, but it's just really usually quite difficult to see with this sort of incidence plot. So what we did next was to analyze all of the 300 species we had, and we created this plot, which is again super difficult to understand. So I'm going to go slowly through it as well. So this is on the x-axis proportion of forest cover. Here is distance to range edge. The lines are the predicted incidence. The colors represent the line just to make it pretty. And if the hue kind of like if it's darker, it's because we've got more data. So when you've got like these white bits here, it's because we don't have much data for that. And what we found was that, yes, there was a positive effect of forest cover on incidence in general, like overall. There was a positive effect of distance to range edge, but there was a negative interaction between these two variables. And what does this mean? So the best way to interpret this is if you kind of fix, for instance, let's say at 100 kilometers from the range edge, and then you look at how incidence changes from 0% of forest cover to 100%. So at 0% forest cover, or close to it, the predicted incidence for the summary predicted incidence would be 0.1. But if at 100% forest cover, it would be 0.3. So it increases, right? And this is true up until, think about 850, 860 kilometers from the range edge. So in general, if you increase forest cover, then the incidence of those populations would increase. So which is kind of the case that we see for the woodpecker here. Now, if you look above this, in the part above the 860 kilometers, what happens is that then, let's say we fix here at 1600 kilometers, then it starts out at 0.6 of incidence, but then here, then we get to 0.3. So the incidence decreases with forest cover, which is exactly what we're seeing here for this woodpecker. So it completely flips around. And what's also interesting is that although distance to range edge always had a positive effect on incidence, you see that it was only at lower levels of forest cover that it really changed dramatically. So what it suggests is that habitat loss is also allowing species to become far more abundant, or increase their incidence far more than they've ever been allowed to, because areas with much more with 100% forest cover, then the highest incidence that they would reach is 0.4. Right. So this is all very interesting. Doesn't actually explain why. What's going on? What is it about these edges that is interesting, that it's driving such changes in species responses to habitat loss? And so one of the things that we did in this paper was that we only measured the distance to what we called soft range edges. So a species, for instance, like this one, that would have this kind of range, it would have what we could call a hard range edge, which is like an edge that it's defined by the coastline, for instance, or like a very abrupt, like a big river or potentially a mountain range, something that would just stop the range right there. But the conditions don't actually change that much from the coastline inwards. But what happens at the soft ranges, the soft edges, is slightly different, because here is just like a slow transition. So imagine that, for instance, here we might have a savanna. So it's just a slow transition from a humid forest to then becoming a drier habitat. So for these populations, it's probably quite different, the impact that they have from these hard edges than they have to the soft range edges. And we had only measured the distance to the soft range edges, but all of the other studies that we've seen so far had measured distance to all of the edges, not separating them. So we were wondering, okay, so are all of these range edges the same? Do they all have the same effect? And also we were wondering, okay, so to which extent our results are applicable to the rest of the world, right? So again, and I apologize to just do stuff on birds. Birds are nice, but let's face it, there's lots of other species. But the thing about birds is that they have really good range maps. That is a problem. And I mean, or a solution. And for the other groups, it's not nearly as good. So Natasha is doing a PhD with me. Natasha is frankly one of the cleverest person I've ever met in my life. So this was her master's. So she put together several databases, putting together up to 116 studies, which reached about 7,500 sites, because there's lots of sites within each study, with I think 2,500 species, something like that, across the world. So we tested these questions on this big data set to see, okay, so the results that we saw for the Atlantic forest, are they applicable to the rest of the world? And how can we tease apart this whole idea of the ranges? And so, yes, the results are applicable. We found exactly the same results. So, but it's only if we measure the distance to the soft range edges. Now here we changed to limits because the reviewer had an issue with us calling it edges. So I'll keep saying it edges, but here you see all about limits. So if we measure the distance from the soft limits, you see again that there is this really strong effect of forest cover, strong effect of distance to range edge, but there was this negative interaction such that the relationship flips again. And Natasha put it in two different ways as well. So these two graphs represent exactly the same thing. But so for instance, if you're 200 kilometers from the range edge, you're likely to increase in incidence. If you're at 1,500 kilometers from the range edge, you're likely to decrease in incidence. Hard limits, when we just measured the distance to the hard limits, we did not see the same relationship. We had to subset the data to make sense, and that's why the numbers here are not exactly the same. But basically also this model had much lower support. So it suggested to us that really whatever is mediating the impacts, the species sensitivity to habitat loss is just what's happening at the soft edges and not the hard ones. We also looked into equator wood and pole wood. And the reason for that is that, again, since Darwin, there's this idea that ranges that, so the equator wood, kind of the side of the ranges, are more likely to be limited by biotic interactions, while the pole woods are more likely to be limited by abiotic interactions. So, just basically it's cold. So here the results are a little bit mixed. So the statistical results for both of these models were exactly the same. There was no higher kind of support for one or another. When you look at the graphs, you can see they're slightly different, but obviously there's huge confidence intervals, so we shouldn't be looking much into that. But what Natasha did next was then to say, okay, so let's put all of these factors in and let's see what is it that could be driving all of this variation in species sensitivity. And sorry, hang on, yeah. And then this was also really quite interesting because so these are results that literally have just come out. So we looked at lots of different variables, and including then we started saying, okay, so what if the distance to the range edge is actually the distance to the cold temperatures that these species are, the coldest temperature that these species can sustain, or the hottest temperatures that these species can sustain, right? And what we found was that actually the main factor determining this is the warm limit. So basically, hang on, let me see. So, right, yeah, sorry. This was a slide that I put just in the last minute, and it got a little bit confusing here. But it led us to wonder to which extent could temperature be driving all of these effects of range edges that we've looked into now. So to get into that idea, we have to remember the whole business of thermal responses. So all species have a thermal response curve like this one, and basically what happens is that there is going to be a temperature that it's your optimum, and there's going to be a temperature that below that you're going to die, above that you're going to die, right? So that exists for everyone, for every single species, but it is also very different between different species. So if we think about people as species, for instance, I was born and raised in Brazil. My optimal temperature is 25 degrees. That's where the area where I'm happiest. I can handle 35, but below 10 I'm unhappy, pretty unhappy. Now my son was born in the UK and has lived in the UK his entire life. He's proper English now. So his thermal optimal is 15 degrees. When it's 5 degrees, I have to yell at him to put some trousers. But by 28, he's kind of all going up in hives, and he just cannot handle it. So if you think about that, so it doesn't matter what is you're going to be your thermal maxima. Every species will have them. It just depends on how far you are from that, from this place. So what we did was to go back to the idea, okay, so we know we have these ranges. Can we then overlay the temperature that exists in these ranges and see how far those populations are from the maximum temperature that they experience within the range? So, for instance, if we go back again to this population here, it's pretty south in a way, so it's pretty low down. So let's say the maximum temperature that it experiences 40 degrees, and let's say the maximum temperature that exists here is like 25. So then this population is 15 degrees from its thermal maximum, while this population here would be probably, let's say, 39 degrees. So it's only 1 degree from its thermal maximum. So this is how we measured. So we looked at I'm never going to be able to explain, because it's the worst explanation in the world when you read the bioclimatic. It's the five, the bioclim. I can't remember, but it's basically it's the hottest. It's an average of the hottest temperature of the hottest month. So that's what we were looking at. Right. So this is what we calculated the thermal maxima for all of these species, for all of the 2,500 species we had. So this was basically for all of the data set that we had. Again, it's slightly fewer studies because we trimmed some because of issues they had. But and we had 2,260 species. So as you can see here, most of the species had their thermal maxima at about 35 to 38 degrees. Okay, some species, kind of like my sun, 25. Some can head all the way to 50, impressive. But most of them are kind of there, right? And it's probably because most of the data we had was from the neotropics. So, so most of the species were from there. So this probably determined a bit. There were few species from the Nearctic, otherwise we probably would have potentially more species with a lower thermal limit. But here's just to show as well that we did have quite a nice variation of forest cover across all of these studies. So it was, yeah, it's a neat study. So first thing we did was to look again. Okay, so do we find that species sensitivity to habitat loss changes with the distance to the thermal maximum? So here is forest cover proportion, varying from 0 to 100%. And here's the incidence. So when a species is close to the warm limit, so let's say the maximum they can handle is 40 degrees and they're in an area that does 39 degrees, they tend to increase with forest cover. The incidence tends to increase with forest cover. But now if they're far from the warm limit, then it tends to decrease with forest cover. Now you can say, oh, but that's likely to be just, differences between species. So that's like an interspecific variation rather than intra. So this is what we did next, was to look at, so we calculated the derivative of the species responses with respect to forest cover. So basically, if you've got a positive number here, it's because you've got this sort of relationship, a positive effect of increasing incidence and forest cover. Sorry, the other way around. And if you've got a negative value, then you've got a negative effect of increasing forest cover on incidence. So you've got this kind of haystack here. It's obviously quite messy, as all kind of really big data sets are. But there is quite a strong trend in overall that populations in areas where they are quite close to their thermal maxima will always respond positively to forest cover, and populations that are further away from the thermal maxima will tend to respond negatively. What's interesting here as well is that the area where it switches, it's about 15 degrees from the thermal maximum. And this was just something that I noticed today, actually. This paper is still in review, so we don't know what's gonna happen. But what we did next was to say, okay, so let's then measure the thermal responses for each of those species. So we standardize them by the distance to the climatic maximum. And again, it's a mess because it's a lot of species. It's always going to be a mess. But what we found is that roughly around most species, the highest incidence you get, it's about 15 degrees from the thermal maximum. And then, it's, yeah. So basically, this is what happens. So all species all populations are responding to temperature in a way that they tend to have their maximum maximum incidence at about 15 degrees from the thermal maximum. So what this means is that if you are in a place that's quite cold for your species, right? So let's say, then again, you know, the example, I'm from Brazil, I'm in the UK, it's quite cold for me. If the temperature goes up a little bit, I'm going to benefit by it. So the populations will increase. If you're in a place that it's already quite hot and the temperature goes up, then what's going to happen is that your incidence is going to go down. Why is this important? The reason why this is important is because habitat loss increases temperature by 4 degrees on average. So basically, we did a microclimatic modeling for all of these areas, and we estimated what would be the microclimate temperature in each of these sites, considering that they have more forest or less forest. And what we found was that if you reduce from 90% forest cover all the way down to 5% forest cover, you increase the temperature by 4 degrees. And anyone who's been in forest can tell that, right? You go into the forest, it's suddenly cool. You step outside the forest, it's hot. It's simple as that, right? And all of those animals are experiencing that as well. So what's going on there? So what happens is, if you happen to be in an area of your range that it's a bit cool for you, and then there's deforestation, this might not be that bad for you. You know, you're still going to be living in smaller patches. They are going to be hotter. It's going to be positive. It's going to positively influence those populations. But now, if you're in an area that you're already a little bit uncomfortable, it's already a little bit too hot for that population, and then you throw deforestation on the top of it and you increase the temperature by quite a lot, then the incidence is going to decrease even more. So this is basically the results that I was showing you before, that could explain why we have these completely different responses to forest cover, to deforestation, whatever way you'd like to say it, for the same species, because it all depends on how comfortable they already are in their own environment. So we've created this kind of graph to kind of help explain, because this is such an unintuitive and complicated result. But basically, deforestation always increases the temperature by 4 degrees in all places. So this is something that we found, that in all areas of the world, there's always this increase. But obviously, if you're in a hot place and you deforest, then you go from 36 degrees to 40 degrees, while if you're in a colder place, it goes from 15 to 19. Now, for a population, you know, that occurs, let's say, throughout South America, and if this population here, right at the north, that population is in an area that is already going to be quite hot for the species. So that population is likely to be much more abundant, or have a higher incidence in more forested areas. While if we go all the way down south, then this population may actually benefit from some level of deforestation. So complicated, huh? Right. So, and again, as a Brazilian, I find interesting the sort of differences in culture that we have. So one of the sayings that we have in Brazil is that, you know, when it's something really good, you want to have sombra y água fresca, you know, which is like shade and fresh water, because it's just, you know, it's what you like as someone who lives in the tropics. But now in the UK, I was surprised when it was like a place in the sun. Like, well, a place in the sun? I don't want a place in the sun. I want a place in the shade. But it shows the differences that, you know, depending on where we are, we may look for different conditions because the environment affects us in different ways. So to conclude, so there's quite a few things to take out, but like what my main kind of 3 points is, so my generalist species may not be maybe your specialist species. So there's a lot of variation in sensitivity. So just something to be aware of. And we found as much variation within a species in responses to forest cover as we found between different species. The effects of both climate and habitat change on populations are mediated by temperature. And yes, there are lots of other things that matter as well, but temperature is one of the most important. And because of that, whenever we start, if we want to predict how biodiversity is going to change in the future, we have to look at temperature, both as to understand how species are going to respond to habitat change, but also to climate change. And we have to kind of start building models that are much more kind of spatially explicit and kind of take into account these thermal responses. So that's it for me. Thank you. I have no idea how long I went for. That seems to be all right. And I'm happy to take questions if you have them. And I'm sorry there was some jumps, but you should never add a slide in the train ride. That's my lesson for today. Thank you.
Okay, thank you very much, Cristina, for your talk. So we open the floor for some questions if you have.
Hi, thank you so much for the talk. I'm curious, I have two questions, very specific. So I would like to know what you said about which traits you use to measure the species sensitivity. And from the image that you showed, I noticed that I think none of those species are migratory, but I don't know if you took that into account or are you planning to?
Right. We use all of the traits that exist in AvoNet. Yeah, pretty much. So no, like we tried, and I think the problem with traits is that they are again fixed at the species level, right? So it is quite possible that if we had intraspecific information, we'd find that populations perhaps more, closer to the thermal maxima may have different traits. They may be smaller, they may be larger. There's, they like the body size, So I think if I were to kind of move forward with traits, I'd probably be much more interested in looking at how they vary across the range. Your second question was about, oh, sorry, I forgot your second question. Migratory birds. I think we removed them. And I think, yeah, so for the second paper that's still in review, that would definitely remove them because someone said we should. But the first one, I can't remember if we did it or not. I can't remember. Yeah, sorry for that. But there was a good point about the reason why we would have to. I think it was the difficulty measuring. So for instance, it may be that the species, the thermal maximum for that species is 40 degrees in Russia, but the species is not in Russia at that time. So to not deal with that, we kind of remove them. Yeah.
I'm guessing they have kind of plasticity towards temperature, something.
Yeah, they may be. Yeah, yeah, yeah.
Just intervening here related to the traits. So we work a lot in functional ecology. My question is kind of just broad, is it, do you think that functional ecology is now, as it stands, doing actually the job for actually quantifying properly and understanding biodiversity responses to environmental change?
So and it's not because I'm here, but I'll say that I think plants are doing a far better job than animals. And it could, like, if you completely deforest an area, you're going to lose a carbon, right? Like, I mean, there's no way about it. Like, but the problem is if you remove some of the frugivorous birds, you're still going to have the primates that are going to do that frugivory. You still so I think the issue with functional ecology within animals is that we're so narrow-minded that we only focus on one group, and it's partially because it is hard. I mean, like, who has the expertise and data to do all of these groups, right? But for instance, one of the things that we've seen before is that some of the roles that birds have in like eating insects, if you kind of exclude them, the spiders start taking on those roles, you know. So the difficulty is that we have to be more holistic than what we're doing. And because, again, you know, if you're working with birds, you use Avonet, which is, you know, the big database. But what's a database for spiders? I don't know. And, you know, like we don't even know who it's what. So I think that's one of the problems. So, and I think the other problem as well is that the traits we use for animals, and I don't know whether that's true for plants as well, but they're all just kind of collected on 3 or 5 individuals, and then you say the whole species is like this. It's not, is it? It's really not. And then we ignore juveniles, we ignore male, female, we ignore everything. So I think there could be more to it, but I don't think we're currently doing a good job. I think it's far more complex than we like to say it is.
Thank you. Damn, I loved it. And it's so nice to see how you've been coming full circle through your career, starting with local scale landscape ecology, looking at edges, and then adding a layer of that +1 with Rob looking at temperature and micro habitat conditions and so on. And now kind of applying the same or similar thinking, but at such a macro scale. Yeah, amazing. But I was just wondering, so, and knowing that you've been doing some work with bioacoustics as well, temperature affects activity as well. And we did a paper with Katarina that is there, in which we were looking at activity patterns of bats in this island where they don't have predators. And we came to realize that there's a diurnal bat there. But the diurnal bat, and this is quite common in places where you don't have birds of prey, but this diurnal bat is only diurnal in the forest, is not diurnal in open spaces. And what we argue in the paper is that it's temperature. And there's quite a lot of literature saying that bats are especially sensitive to differences in temperature and whatnot. But I guess that should be more widespread as well. So not only it impacts incidence and abundance and so on, it will impact activity patterns across the board. And now we have the tools to look into that. So I was just wondering what your thoughts might be on that.
Absolutely, and particularly for insects, right? It's the basis of all insect activity as well. So it's going to be temperature, and then those animals are going to, like lots of the vertebrates are going to be eating the insects. So it's hard to know exactly whether they're responding directly to the temperature or whether it's a response fed through the insects as well. But I would imagine a lot of it is also, like, we know that, for instance, bats in the UK, they hibernate in winter, so they're obviously responding directly to the temperature as well. I mean, I don't really have, I think it affects everything. And I have wondered, because, I mean, you're probably going to correct me, but I'm going to say something, you're going to be angry. So, but like,
Birds are better than bats.
birds, well, so bats, so my understanding of the literature, particularly in the neotropics, is that bats are less likely to be highly sensitive to deforestation. Oh, I knew. But I wonder whether perhaps the fact that they're nocturnal is something that changes that a bit as well, because they are experiencing different temperatures. So that is something that we obviously would have to take into account, and perhaps, you know, for night birds as well, that would be also the case. But I do wonder whether this is something that needs to be taken into account. And then if you're looking like at small mammals, then it's yet a different axis, because then you're talking about the temperature in the soil, right? Which is far more constant than the air temperature. But it is. Yeah.
Thank you so much for this amazing talk. My question was more related to your result about the species that were further away from their warm limit, and why they had less incidence with decrease in forest cover. So if you can like say why that happened. And also just wanted to clarify, did you use like mostly forest specialists bird species? Did you have a filter or something?
We use all species.
Ah, okay. Okay.
Yeah, we use all species. So your question was why were they were being benefited, but
with increase in forest cover.
because it's colder, makes it even colder for them. So yeah, so these would be species populations that would, you know, they still exist in the continuous forest, let's put it this way, like, you know, comparing like a very large forest with a smaller one. So they still be present there, but it's just a little bit too cold for them. So then if you deforest that to, I don't know, 10% forest cover, then that forest is going to be a lot hotter than the very large bit of forest. So for that species, that is just, it was just free, you know, a little bit too cold for them. So that's going to be better for them. So they're more likely to be seen in these smaller patches or in the more deforested area. Yeah.
Yeah, thank you very much for the talk. It was very interesting. I was just wondering if you've ever considered using this kind of data with sort of future climate models to see if that could help inform sort of which areas to protect or which populations are most vulnerable, or whether that's a viable thing you're looking into.
That's a very good question. That's Natasha's chapter number 3. So, so that's what she just sent me some of the results yesterday. And yes, that's exactly what we're trying to do. But it is all very early stages, right? I can't say much. I think so. There's a few problems. One is that so we're trying to figure out the approach. If you control for everything and then just change the temperature, you can have a much better, much more reliable idea of what's happening. Now, that doesn't extrapolate to the rest of the world. So you're just kind of stuck with those kind of areas that you're studying. So that's kind of what we're trying to figure out, because if you extrapolate, then you're going to lose, it's going to become a lot less robust. So we're trying to figure out what to do. But here's what I found was very interesting. By increasing the temperature, because the vast majority of populations are in areas that are more likely to be colder for them, we would expect an increase in an incidence. I wasn't expecting that. And I think it is something that I've noticed with the narrative of climate change, though, that we always focusing on the species that are right at the edge. So like, yeah, I think the polar bear is in a dire situation. No, is the polar bear? No, yeah, probably. So Amazon forest, right? That's probably the best. Like it's pretty hot already. Like it's really bloody hot. So if it goes to 50 degrees, it's gonna be beyond the physiological limits of most species. So we focus a lot on that, but we stop to think a little bit about all of the species all of the populations that are in the colder area. Like they're not going to be that heavily impacted. It's going to be good for them. So I have to think I mean, we literally just got the results 2 days ago. So I have to think what's going to come out of it. It was an incredible amount of work, and we're still not sure whether the approach is quite correct. But this is exactly what we're trying to do, kind of trying to change forest cover, increasing or decreasing, change temperatures, and see how we can predict biodiversity change. It's hard, much harder than I thought it would be.
Yeah, very exciting though.
It is. And yeah.
You mentioned that temperature was definitely the one that was most important in driving this trend, but I was wondering if you've tested other factors that change with edges and how that would impact.
We tested lots of factors. Yeah, just about every single factor that you could think of. Yeah, so we've created this kind of random forest model that you can just, because lots of these factors are correlated, so that's the problem. So you have to kind of take that into account, and then what we were able to find is that, yes, there are some other factors that are important. I think latitude is important, elevation to some level, range size. And there were some other macroecological factors that were important, which is quite interesting as well. There were very few species traits that were important that mediated any of those responses. But the distance to the warm limit was the most important one. We also looked at the distance to the cold limit, but that wasn't nearly as important as the warm limit, although it does have some influence.
Thank you so much for the talk. I thought it was really well explained, so a layman like me could follow. There was one thing that I don't quite understand yet. I think not because you didn't explain it, but maybe I missed it. So you showed us this this kind of like normal distribution, like this performance curve, where you said, so if it's beyond the optimal limit, like it's too hot, then a species will perform better if it gets colder, and vice versa. And then you showed us this map of South America where species closer to the equator would benefit from deforestation because it gets colder then in that area, or like the temperature, they would benefit from temperature decrease in their range. And I'm not sure if that's like a bit of like a circular reasoning where you say like initially you use the data that shows that species benefit when it gets colder in hot areas, and then you show that the exact same thing kind of. So I was wondering whether it's necessary to use some other gradient, let's say temperature over like an altitudinal cline or something, to see whether the same pattern can be found there.
Yeah, no, so this issue of circularity is one that we discussed a lot, actually. So it's not the same measure that we're using. So for instance, when I showed the map of South America and where the range of the species was, we said, okay, so if the population is here and so it's all about we don't have to necessarily look at the distance in space, right? So we can just say, okay, so across the range, the hottest temperature is 40 degrees. And the population is in a place where the hottest temperature is 25 degrees. So it's 15 degrees of distance. But when we went to calculate the species thermal curves, we used the microclimatic temperatures. So it's, that's all right. Yeah, the answer is wrong. So we use the microclimatic temperatures. So it wasn't exactly the same temperatures looking at, like the same measure as looking at the range. So we basically ran a model where you say, okay, so this is the area, this is kind of like, there is, the macroclimatic temperature there, but this is the kind of the amount of forest, this is the type of forest that there is there. And there's lots of different kind of measures to try to predict what is the actual temperature that is occurring there. So when we created those curves, that's how we created with those other temperatures. But I think essentially it's kind of like, it's a little bit intuitive. It's complicated, but it is intuitive in the sense that if you're in a hot place, you want to go to a cooler place. If you're in a cooler place, you want to go to a slightly hotter place. So it's basically down to that. And that is what the deforestation is doing. So if you're in a really hot place and the forest is reducing your temperature, you want to be right in the forest. You want to be in the coolest place possible because that is already comfortable for you. So my son in that forest would go right in the middle into the forest, where I could probably be at the edge. While, in a cooler place, and the species is a little bit, like the population is just a little bit uncomfortable, like in a physiological sense, is it just a bit too cool, then they want to be in a slightly warmer place. So it's kind of like thinking forests are basically creating the microclimate for them. Does it make sense? Yeah.
Thank you. Yeah, thank you very much. I wondered, for those that are well within their thermal limit, like, is there a point at which habitat loss becomes important? Because presumably the line just wouldn't keep going down. Once you have zero forest cover, they won't have anything to, well, any resources, place to stay, raise children, etc. Did you see a point where like there was a sharp?
We didn't look for it. That's the short answer. I'd say, yeah. So most of these studies were conducted inside the forests. So even though we looked at a large gradient of deforestation, they were still in the forest. So we were looking for species inside the forest. So it's, I suppose it's the difference between looking at a forest patch of 0.5 hectare and 1,000 hectares, or 10,000 hectares. Yeah, they would probably lose their habitat altogether. But having said that, one of the kind of like the whole discussion that I had with the Argentinian guy was that exactly that, like, the species that I could only see in my primary forests, he was seeing in the gardens outside the forest. So we were like, well, how do you put this together?
Yeah.
So I mean, I'm not saying this, let's just deforest everything. That's absolutely not what I'm saying. But I think species are far more flexible and plastic than we also think they are.
If I might be cheeky and ask a very quick second question, like, forests are obviously very good at insulating. So would you expect these results to sort of switch if you looked at places further towards the poles, where perhaps they're in fact keeping the temperature above their lower thermal limit?
We looked at that in the paper. I can't remember the answer. I think the results were roughly the same, a little, but we have fewer data points, so it was a little bit weak, a little bit less interesting. It's not something that I can definitely say, yeah, in the polls it's going to be like this, because there were just fewer data points, but we looked. I can't remember.
Thank you.
Yeah, thanks very much. I was curious about the resource question as well, and wondering if there's a certain point past which, if you lose enough forest, you've lost enough resources, that you'd see a trend. And I think part of the way of thinking about that might be, how is incidence measured, and how is that different to population size, or some of like the more kind of conventional measures of abundance that we would think of?
Yeah, that's a good question. So it was just basic presence, not absence. Partially because so many of these studies were using point counts. You can't measure abundance using point counts. You can't tell whether it's different individuals or not. But I have my reservations with abundance measures, because I think abundance is often so much more complicated. Like it's just so much harder to find trends that make sense, and so I think for these sorts of data sets, I would much prefer using a presence absence than abundance. But the studies I've done where we did have a good measure of abundance, like for instance using mist nets, where you can tag species in individuals, then we found very strong correlation between abundance and incidence. So yeah, but I think, yeah, so it's, I mean, with these sorts of with these sorts of really large studies, it's hard to get into the details, because we're also talking about nearly 2,500 species that will be eating completely different things, using completely different resources. So it's hard to kind of really look into the details. I think there's one final question there, and yeah.
Yeah, very interesting talk and discussion. And I was during the talk, I got the thought, like, yeah, you're looking at niches, and the distance to the core of the niche, especially on a large scale, climatic variables are probably very important. And then you arrived at climate, and the climatic niche for one species probably correlates a lot with the species which in the same area, but that doesn't. They likely have more or less slightly climatic niches as well. So if the temperature changes, it doesn't change the same for everybody. And biodiversity wise, I know the connection between species comes under stress. So while I wonder if in the middle or long term you'll see the same things, or if then things might break down because the connections between species become under more stress, so the populations might become more vulnerable to these changes over time.
Right. That's a good question. I don't know. Haven't really thought much about this. Yeah. So species will all be interconnected. Each of them are going to be at a different part of their thermal limit, the thermal performance curve. It's hard to know, because then we really would need to have this kind of better idea of how strong those interactions are and how important they are, right? Because they can be strong, but they may not necessarily be crucial in the sense that, yeah, let's say I have a very strong interaction with her here, but then if I move, the rest of my population has a very strong interaction with him. So it may be that all of that is very flexible and plastic. We don't know that. And obviously, again, I would say that we have to look at that on a much more holistic sense. It's not just looking at birds, it's birds' interactions with plants, with the insects, with the mammals, with everything, right? Because they are being positively and negatively interacted by all of these different groups. So, which is why we think there is a role for biotic interactions in this. So the result that we found for edges, range edges, is mostly explained by the temperature, but not fully. There's still a little bit that's different, and I would imagine that that's down to biotic interactions, but with who? I don't know. Because again, it could be with so many different species. It could be anybody. Could be anybody. It could be your predator, which is a snake. It could be your food, which is an insect. It could be anything. So that's the complicated part. But I do think biotic interactions still play a role. It's just that it's hard to measure.
Okay. Cristina, thank you very much for this fantastic seminar. So we'll have drinks in the atmosphere room just around the corner if you want to join us for this and continue chatting with the speaker.
It's a great name for a room.